We're in the cell transport. We will look at how the molecules move through the cell membrane, in and out. First, diffusion. Every molecule naturally goes from the high concentration to the low concentration area. It's called simple diffusion because every molecules move. So eventually, they will become the same concentration. Like you accidentally break a bottle of perfume, or you open a bottle of perfume, the perfume molecules will go from the high concentration to the low concentration area. And eventually the whole room will be the same concentration. That's diffusion. So why does diffusion occur? Because every molecules move as long as you have temperature. The temperature is not absolute 0 degree, which is minus 273 degree. Every molecule move. So they have kinetic energy, they will move. And that's the driving force for the molecule to move from the high concentration to the low concentration area. So if you have a body part, they have a high concentration, and these molecules, they will go from high to low. So what affect the diffusion rate? 4 factors. The first one is the size of the molecule. Apparently, smaller molecule run faster, so size is a factor. Second one is temperature. When you start to increase the temperature, the diffusion rate increase because they have more kinetic energy. The third one is the concentration gradient. Concentration gradient is a fancy word to say the concentration difference between 2 areas. And if there is no concentration gradient, the particle don't have to move. So the particle will move from high concentration to the low concentration area. We say they follow the concentration gradient. And the last one is distance. Diffusion works pretty well for short distance. It does not work well for long distance. So these are the 4 factors. Let's review them. So first, molecular size. If the molecule is bigger, like these experiments you see in AP1, you have 2 dyes. The lighter dye gonna run faster. You will see gradually diffuse out in the argon media. Argon is mainly water, 99% water, so it will diffuse out. And the blue dye, which is heavier, it will diffuse, it will move slower. So bigger molecules run slower. When you increase the molecular size, molecular weight, the diffusion rate decreases. The second one is the temperature. So temperature, when you increase the temperature, the diffusion rate increase because the molecule can run faster. And when you decrease the temperature, the molecule runs slower. The third one is the concentration gradient. That's the concentration difference between the high concentration area and the low concentration area. When you increase the concentration gradient and the diffusion rate increase, and diffusion stop when there's no concentration gradient. The last one is the distance. When you increase the distance, diffusion rate decrease because diffusion works pretty well for short distance. Like you break a bottle of perfume, your room smells like perfume, but your whole house probably won't because the diffusion rate increase. So diffusion works pretty well for short distance. So these are the factors affect diffusion. Now let's look at the water diffusion. Water still goes from high water to low water, but water is a very important molecule in biology. So we never say water diffusion, we call it osmosis. So osmosis is water diffusion. Like you have an artificial cell membrane, and you have high water, and on the right side, you have low water. So water will go from high water to low water, exactly the same as any molecules diffuse from high concentration to the low concentration area. However, students have more difficult to picture water diffusion because water in a biological system is always being used as a solvent. So I tell students to think about water. Water goes from low solute, which is on the left side, to the high solute. This solute is sugar, to the right side to dilute the solute. So the solute, like the sugar, the green ball here, you still want to go from right to left because every molecule want to follow its concentration gradient. But in this scenario, it can't because apparently sugar could not go through the cell membrane without helper. So the sugar could not go. And in this case, if the sugar could not go, water will go to dilute the high sugar. So water diffusion is osmosis, and water goes from low solute to the high solute. Or you can think water goes from high water to low water, even though some students find it's more confusing. So I tell my students, say water goes from low solute to high solute to dilute the solute. So that's osmosis. Osmosis is water movement. And water go to dilute the solute. And once they dissolve, the solvent, the solution becomes solvent. The solvent is the liquid dilute, but in the biological system, we always use water. So water is a solvent. And when you put solute into solvent, now you have solution, like you put salt into water. That's a salt solution. Okay, now let's look at the particle, those solute. Not every solute, not every particle can free go through the cell membrane because in the previous lecture, we talk about, okay, because the cell membrane is mainly made of phospholipid and a lot of molecules could not go through phospholipid. They could not diffuse through the cell membrane. So we call them non-penetrating particle. Like a lot of them, salt, ions, glucose, amino acid, salt dissolved in water become ions. So these are the non-penetrating particle, means they could not go through the cell membrane without helper. In that case, okay, say you have a cell. You have a cell. You put the cell into a solution. Outside is 100% water, pure water. What's gonna happen? What gonna happen is inside the cell you have some sugar, and you have water because that's the ICF. And say ECF is 100% water. The ICF is only 6% water. So what's gonna happen? And I told you water will always go to dilute the solute. So inside the cell, you have sugar. It will try to diffuse out, but it can't because of cell membrane. In this case, okay, sugar could not diffuse out. The only thing can move is water. Water will diffuse in, that's osmosis. Water will move in to dilute the sugar. It will keep moving in until the inside and outside, there's no concentration gradient, which is 0% glucose. So water will keep moving in until the cells swell and burst. So what happened to the cell is the cell will swell and burst. And we have a name of this solution called hypotonic solution. And another scenario, okay, in this case, the cells inside have less solute than outside. So outside have 10% sodium chloride, 90% water, and inside have 100% water. In this case, what's gonna happen? In this case, okay, water will move out. Water will move out. You can say the salt want to move in, but the salt is non-penetrating. So the salt could not move in, could not move through the cell membrane. So what happens is actually water will move out to dilute the salt. Water will keep moving out until inside become 10% salt. So water will move out. When the water move out, the cell will shrink. So we call this solution hypertonic solution. Hypertonic solution make the cell shrink. And that's the concept of tonicity. Ideally, you want the solution you put the red blood cell in would be exactly the same concentration as the ICF. So water won't move in, move out because they are the same concentration. In that case, the cell maintain the same shape, and we say that solution is isotonic solution. So isotonic solutions make the cell stay the same shape. Then what if you put a red blood cell into a solution and then the cell swell? The cell swell because inside concentration is higher than the outside. Outside is lower. So water have to move in to dilute the solute inside. Water move in, the cell swell. So we say the outside concentration is too low. Too low means hypotonic. So hypotonic solution make the cell swell. Or a big word called hemolyze. Hemolyze, hemolyze. Hemolyze is a big word to say the cell burst. Hemolyze, Hemolyze. So hypotonic solution, hemolyze the red blood cell. And what if you put the red blood cell into a solution and the cell shrink? The cell shrink because the outside concentration, solute concentration is too high. Because it's too high, so water have to move out to dilute the solute outside. Water move out, the cell shrink, or we say crenate, C-R-E-N-A-T-E, crenate. And this concentration is too high. So too high means hypertonic solution, crenate, make the crenate red blood cell or make the red blood cell shrink. So hypertonic solutions make the cell shrink. We have three kinds of tonicity. So tonicity only have three. Hypertonic solution, crenate red blood cell, make the cell shrink. Isotonic, make the cell stay in the same shape. And hypotonic solution, make the red blood cell swell or burst or hemolyze. And apparently you can change your ECF environment, make this, or this happen, because every day we lose water. You sweat, you urinate, you breathe out water. So every day you lose water. It means that's why doctor asks you to drink water about 1.6 liter per day, because if you don't drink water, gradually your ECF, extracellular fluid, will become a hypertonic because you gradually lose water and your cell will shrink and you become unhealthy. So you need to drink water, make it go back to isotonic. That's the environment we want to stay. However, now you know the ECF, if it's a hypotonic, it actually will make this water move into the cell, make the cell swell or burst or hemolyzed. So if you drink too much water too quickly, actually it's not healthy because the water can move in to the cell too quickly and the cell will burst and red blood cell die. And the worst scenario, you get sick or you die. It's called water intoxication. It happens about 15 years ago when this lady, she participated in the contest, which sponsored by the local radio station, asked them to drink as much water as you can and hold your pee to win the shiny Wii. About 15 years ago, a shiny Wii was a very, very good toy. And this lady, she won. She drank 2 liters of water in 10 minutes. But 2 liters water in 10 minutes is too much. So she turned her ECF, extracellular fluid, into a hypotonic solution, and which make her red blood cell burst, hemolyzed. And when you have too many red blood cell burst, you don't have enough oxygen carry ability. And eventually she dies. So that's called water intoxication. And every big hospital, they have the water intoxication lab to treat people. They have water poisoning. And a lot of patients, actually, they're pretty healthy patients because they care about their body. They want to drink water. But actually, a lot of people don't know you can drink too much water to turn your ECF into a hypotonic, which will make your red blood cells swell. It's not healthy as well. Okay, let's stop here.